not just activation energy. Yeah, exactly. Remember that an oxidation reaction is just a half reaction. If I'm giving something to another person, if that other person isn't around, I can't give it away. In order to have an oxidation reaction, you simultaneously must have a reduction reaction. You can't have one without the other. Something gives away an electron, something else has to take it. Therefore, the combined reaction is called a redox reaction. Okay? You need both to occur simultaneously. Now, in biology, we can't move electrons. Okay? Because we don't have free moving electrons, right? You're not made of metals, right? If you were a living organism that was made out of copper, yeah, we could move electrons. How do we know that? Because we do it in things like this all the time. In this lamp that's got copper wiring, we move electrons all the time. But in you're a biological organism made of carbon. If you're a carbon existing organism, you don't have free electrons to move around. Okay? So to move electrons in biology, you have to move it as a part of something else. And generally speaking, we move it as part of a hydrogen atom. Because when you move a hydrogen, you're moving a proton and electron. And therefore, you're moving the proton to keep the electron, to keep the charge neutral. But moving hydrogen is equivalent to moving electrons in biology. Okay? So if a substance is oxidized, it's literally moving a hydrogen atom from one molecule to another. Okay, so in biology, a redox reaction is shown by the movement of hydrogens, usually pairs of hydrogens, H2s. Okay, so if you look at this bottom equation, right, for cellular respiration, what have we done? We've moved the hydrogens from glucose off. So glucose becomes carbon dioxide. That's an oxidation. And we've moved the hydrogens onto the oxygen, which becomes water. That's a reduction. Okay. So it's still a redox reaction, but what we literally did is we moved the hydrogens from glucose to oxygen. Okay. So redox reactions in respiration release energy as a breakdown of organic molecules. Okay. We're breaking carbon-carbon bonds. And we're also taking electrons from carbon-hydrogen bonds by removing the hydrogens. Okay. So C6H12O6 becomes carbon dioxide. So the fuel's been oxidized. It lost hydrogens. The electrons move to a more electronegative atom. The most electronegative atom in biology is oxygen. Okay. The oxygen becomes water and the oxygen has been reduced. Okay. So what we're doing is we're coupling these redox reactions. So we're using redox reactions as the exergonic reaction linked to the endergonic reaction of the synthesis of ATP. Okay. That's the energy coupling relationship that exists. Okay. So in cellular respiration, you're using the energy of redox reactions to drive the synthesis of ATP from ADP and phosphate. Okay. This basically summarizes all of cellular respiration in one slide. That's the point. Okay. So now we just go into the details of how we get all of this to work. So if you want, I would suggest you star this particular slide or make note of it somewhere. It's this coupling of oxidation and reduction to ATP synthesis. This is the point. This is exergonic chemical reaction linked to an endergonic one. Okay. Remember all of these clues for differentiating between oxidation and reduction. Oxidation is adding oxygens, removing hydrogen, loss of electrons, releasing energy, which is exergonic. Reduction reactions remove water, add hydrogen, gain electrons, stores energy, and is endergonic. Okay? Effectively, we're doing redox reactions, and the excess energy from exergonic reactions are used to make the ATP. Okay. Anyone not clear on that point? What the point of the redox, because this is the point that most people tend to miss, what the point of the redox reaction itself is. If you're confused, hands up, and I'll try and go over it again. If you're clear, we'll move on. Okay, so in order to make ATP, you need a source of energy. Okay, because taking ADP plus a phosphate and combining them needs energy. So you need an energy source. What we're going to do is we're going to break bonds, okay, in glucose, okay, because the breakdown, digestion of glucose is exergonic, and we're going to use that energy to drive the synthesis of ATP. So normally, what we do in, for example, phosphorylation is we take ATP, convert it to ADP and P, and use that energy to drive endergonic reaction, like say moving something against a concentration gradient, or moving a muscle, or almost any other cellular process. Okay, that's what your cells normally do. But now you need some way to make ATP. So you need an energy source. Well, the energy source is removal of electrons from glucose, transferring them to oxygen, forming water, causes the release of energy. That energy can be captured and used to make ATP. Okay? So we're using the oxidation of glucose to drive the synthesis of ATP. It's still a, a coupling reaction. We're still linking an exergonic chemical reaction to an endergonic one. Okay? We're still using one reaction to drive another, right? The whole, this slide here, right? So instead of thinking about it this way, think of this as glucose going to carbon dioxide and water plus energy. That energy is being used to drive the synthesis of ADP and P to phosphate. That's what's going on in terms of cellular respiration. With ATP, we reverse that. We use ATP up here to drive other chemical reactions in the cell. But when we need to make the ATP, we need something else to do the digestion part. And that's the digestion of glucose to carbon dioxide and water. That energy will be used to make ADP and P into ATP. Okay? Now, how do we actually move electrons in cellular respiration, or in in cells in general? Okay, because we can't, like I said, we can't move electrons, right? You guys are not metals. So what we use, let me just go back one. Ah, okay, so we're going to use a molecule called NAD+. You don't need to know the full name. Okay. The full name is nicotinamide adenine dinucleotide. Okay. But you just need to know it as NAD+. Okay. Based on its name and looking at the components in it, what do you think this molecule is? What kind? Carbohydrate, nucleotide, lipid, or protein. Yeah, it's a nucleic acid. Okay, because we've got an adenine base, we've got a ribose sugar, two phosphates, another ribose sugar. Okay, and then this thing which is nicotinamide. Okay, so nicotinamide, that's this part, adenine, right? And then it's a dinucleotide. Why? Two sugars, two phosphates. Okay, so this is a modified nucleotide, right? How efficient? Build once, use many ways. Okay, now we move electrons in biology by using something, by moving hydrogen atoms. Okay, the molecules that move hydrogens around in biology is called an electron carrier. NAD plus is one of these electron carriers. Okay, so if you take NAD plus and you give it a hydrogen, okay, in other words, you reduce NAD plus, it becomes NADH. Okay, so the oxidized form of this molecule is NAD plus. The reduced form, its accepted electrons or gained a hydrogen atom, is NADH. Okay, you'll notice there are two other names in brackets here. NADH is referred to in three different ways. I'm lazy, I will call it NADH. Okay, but in reality, whenever we're moving electrons in biology, we're moving pairs of hydrogens, H2s. Therefore, this a more accurate way of describing this molecule is NADH2. The most accurate way is NADH plus H plus. Okay, here's what happens. Okay, there's another molecule, FAD, which does the same thing. So there are actually two electron carriers, NAD plus and FAD2 plus. Okay, the hydrogen that NAD plus accepts actually gets bound here. Okay, so this molecule now has electrons. It has the ability to reduce things. It has the electrons. It can now use those electrons to power other things. Okay, so this is NADH. Okay, you don't need to know the structure of this molecule. You need to know what NADH represents. Now, here's the important bit. The reason we call it NADH plus H plus, this molecule as is not balanced. So in fact, there will be a second hydrogen plus associated with this molecule that gets carried around with it. Yeah, it was not paired because we moved an electron, right? And that moving of that electron forms that bond. Okay. So we actually do form a new bond here, and then we also carry another hydrogen ion with it. Okay. So we're now carrying the electron in this molecule as a reduced molecule. Okay. The best way to think of NADH is like a piggy bank. Okay. What we're basically doing is we're taking the electrons in the carbohydrate and we're going to temporarily store the electrons in this electron carrier called NADH. It's like putting money in the bank. When we need to use the power of those electrons later, we'll have access to it. We'll break open this piggy bank and we'll remove the electrons from here. But until we need it, we need someplace to store the electrons. Therefore, they're stored in this molecule as NADH. Okay? So, before I talk about that, I want you to watch this. Let's see if I can find it. Maybe it's under control history. Oh, not history. History, here we go. Okay, I don't know if this will help or not, but I want you guys to take a look at this. I think this is a video.
This video is covering NAD+, and it's made for my 5th year biology class to help them understand this particularly important molecule in preparation for studying cellular respiration. It's really important that you know the full name of the molecule. It's nicotinamide adenine dinucleotide. NAD+ is a carrier molecule. It traps electrons and protons and transfers them to where they're needed in the cell. And that's the best way of describing it. State that it's a carrier molecule, state that it traps electrons and protons, and finally that it transfers those electrons and protons to where they're needed in the cell. It's really important that you know how NAD+ traps the electrons and protons, and where indeed are these electrons and protons coming from? Well, firstly, the electrons and the protons are coming from hydrogen atoms, pairs of hydrogen atoms, and so whenever we consider NAD+ in cellular respiration, we always consider electrons and protons coming from pairs of hydrogen atoms. So the details of this reaction are NAD+ will trap 2 electrons and 1 proton, remember they're coming from hydrogen atoms, and so is converted or is reduced to NADH. And when you compare NAD+ with NADH, we consider NAD+ to be a low energy molecule and NADH to be high energy. To make our equation perfect, we have to account for that one other proton. Remember, we're talking about pairs of hydrogen atoms, 2 electrons and 2 protons, so there's one other proton that needs to be added into the equation. So let's sum up. NAD+ is known as nicotinamide adenine dinucleotide. It's a carrier molecule and it traps electrons and protons and transfers these to where they're needed in the cell. The electrons and the protons are from pairs of hydrogen atoms, and because we're getting ready to study cellular respiration, we're stating that these hydrogen atoms come from glucose or other substrate molecules in cellular respiration. We know that NAD+ is a low energy molecule, and when it traps those electrons and that proton, it turns into NADH, which is a high energy molecule. It's really important that you always remember to write that once NADH transfers those electrons and that proton, that NAD+ is regenerated, all ready to do some more trapping and transferring.
Okay, question. Do you want me to put that little video on Classroom? Yes. Okay. So I'm going to put it here under energy and metabolism because you need that before we get to glycolysis. So let me edit this, add YouTube URL. Now, it probably won't play here. When you click on it, you'll give you an error and tell you to go to YouTube, because these are only designed to be played on the YouTube app, I guess, itself. So I don't know that it'll play on here. Yeah. So watch this. So you have to click watch this video on YouTube in order to watch it. Okay, just so you're aware. Okay. So let's continue on with the ending of this. All right. So cellular respiration basically consists of 4 stages. Okay. We're going to take a look at step 1 next. Okay. The first stage is anaerobic. It does not require oxygen. Okay. So that's what we call glycolysis. Okay. It's the breakdown of glucose without oxygen present. And this is the only step in cellular respiration that occurs in the cytoplasm. Okay, all the other stages occur within the mitochondria. Okay, so there is that distinction between these two stages. Everything that occurs in the mitochondria is considered to be aerobic, although really stage 4 is the only truly aerobic stage. Okay, stage 2 is pyruvate oxidation, stage 3 is Krebs cycle, and then the last stage is what we call the electron transport chain. Okay, and what we're going to be looking at in a fair amount of detail is this equation here. Okay, now I'm going to open up the, you guys don't need to see this, next presentation. So if you go here, Unit 2, Metabolism, Glycolysis. Okay, so I'm going to be going through glycolysis here. There are two handouts that summarize glycolysis, 4A and 4B. You probably want to grab those as well, just so that you have them as a summary of the Google slide. And then afterwards, we'll take a look at the overview of glycolysis. Okay, so we're starting here. Okay, come on, load up. Okay, stage 1 is glycolysis. All right. So by its name, you should be able to tell what glycolysis is, right? The breakdown of glucose. Makes sense. Glyco, meaning to sugar, and lysis, meaning to split. Oh, sorry. Share this tab. There we go. Okay. So we're breaking down glucose, right? Splitting sugar. What we're literally doing is we're taking a 6-carbon glucose molecule, and through a series of 10 enzymatically driven steps, we're going to break it down into 2 molecules of a 3-carbon sugar called pyruvate. Okay? So we're going from glucose to pyruvate. Okay? This is a very old pathway. It's probably about 3.5 billion years old. Okay? It's where energy transfer first evolved. So this is the first form of energy transfer that would have occurred for living organisms. Every single organism on this planet uses it or some form of it. Doesn't matter if you're an archaean, a bacterium, a fungi, a protist, a plant, or a mammal. You're going to use some form of this method of energy transfer. Okay? This is the first pathway that evolved that transfers energy from an organic molecule, okay, to ATP. Okay? And today, it's still the starting point for all cellular respiration. It doesn't matter what organism you're talking about. If it uses cellular respiration, it's going to use glycolysis. Okay? But it is inefficient. Okay? You only generate 2 ATPs for every molecule of glucose. Okay, so let's think about how much energy that is. Okay, so I'm going to put in the chat, and I want you guys to try the calculation. ATP produces 31 kilojoules per mole of energy. That's the amount of energy you get in ATP. Okay, glucose has an energy level of 2831 kilojoules per mole. Okay, so if you're only getting 2 ATP, what's the total amount of energy in glucose What's the percent efficiency? How much energy in the glucose is actually being captured as ATP if you're only getting 2 moles of ATP for every mole of glucose? What's the efficiency? Yeah, it's going to be about 2.2%, right? 61 divided by 2831 times 100. Okay, so the efficiency is incredibly low. Okay, the process also only occurs in the cytosol. Okay, so obviously, for something like us, that's not enough energy to keep us going. Okay, but we still use it as a starting point. Okay, why does the fact that glycolysis only occurs in the cytosol make evolutionary sense? Okay, not all things have mitochondria, but all things have what? All things have a cytoplasm, right? The first, since this is the first energy transfer method, it had to occur in prokaryotes. Prokaryotes don't have mitochondria, but therefore the chemical reactions had to occur without a mitochondria, which means it had to occur in the cytosol. Okay, so from an evolutionary perspective, okay, with the prokaryotes, okay, the first cells would have had no organelles, none whatsoever. They can't. How how did organelles evolve? How did things like mitochondria, um, Golgi bodies, endoplasmic reticulum, how did they evolve? What's the process? Nucleus. Do you remember from grade 11 what we called that? Anyone remember this term? Endosymbiosis, right? So it's the idea that a proto eukaryote would consume another prokaryote, but instead of digesting it, it incorporated that prokaryote into part of its biology. So it's now a symbiotic relationship. Your cells couldn't live without a mitochondria, neither could the mitochondria live without the host cell, right? So it's internal symbiosis, right? So that's how we got organelles. Okay. Remember that we also had an anaerobic atmosphere, right? So the atmosphere itself would have had no free oxygen in the air. If free oxygen became available in the early atmosphere, what would happen to it? What would immediately happen to any free O2? Yeah, it would oxidize, usually combine with iron in the crust and form iron oxide, which is why we have all these iron mines all over the planet. Okay. So back then, we had to grab energy from the organic molecule, but without oxygen present. Okay, so the prokaryotes that evolved glycolysis are the ancestors to all modern life. Okay, and all cells still utilize glycolysis today. Okay, there isn't one single exception. They may not use sugar as the ultimate energy source, could be methane or hydrogen sulfide, but the pathway is still pretty much equivalent to glycolysis. Okay, so the enzymes of glycolysis are pretty much well conserved. If I compared the enzymes of glycolysis in you to the enzymes of glycolysis in a yeast to that in uh, an archaean in a hot spring, they'd basically be the same. The genetic sequences are basically identical. There's very few mutations between them. Okay. Overview. Glycolysis consists of 10 chemical reactions that are going to convert a 6-carbon glucose, which you see up here, to 2 pyruvates, which are 3-carbon, which will be down here. In the process, it produces 4 ATP and 2 NADHs, those electron carriers we talked about. It consumes 2 ATP, therefore we get a net yield of 2 ATPs and 2 NADHs. Okay, so we start with a glucose molecule, go through a couple of enzymatic reactions. ATPs are going to dump, 2 of them are going to attach phosphates to the sugar. So you get up with this molecule. This molecule then splits. So this is equivalent, adding the phosphates to the glucose molecule is equivalent to adding activation energy. This is going to destabilize this glucose molecule. Now that it's destabilized, it can break up into 2 molecules of 2 3-carbon sugars. They're called DHAP and G3P. DHAP is short for dihydroxyacetone phosphate. G3P is glyceraldehyde 3-phosphate. You don't need to know the long forms. You should recognize G3P as the halfway molecule. The reason why G3P is the more important one, for this process, DHAP will be converted to G3P. So at this point, everything happens to DHAP twice, or to G3P twice. So when both of these go through, you're going to have 2 phosphates come in, 2 hydrogens leave, 2 NAD pluses come in and convert to 2 NADHs, 4 ADPs come in, grab the phosphates from the G3P, the 2 G3Ps, and become 4 ATPs, producing 2 pyruvates. Okay? So, the first half of glycolysis is the energy investment. We're putting energy in. So, it's the first half is endergonic. Okay? We're going to invest a little bit of ATP, equivalent to adding activation energy. So we're going to take 2 ATPs and we're going to take those phosphates and attach them to the glucose. And we're going to get 2 ADPs out of it. Okay. By doing that, we're going to get 2 molecules of G3P. Now we get the payoff. Okay. The second half of glycolysis is exergonic. We're going to get a little bit of ATP and a little bit of NADH. So we're going to get 4 ATPs coming off of the G3Ps. Okay. We will have 2 NADHs, and we're going to end up with 2 pyruvates at the end. Okay. So in net, 1 glucose becomes 2 pyruvates and 2 water. 2 ADP plus 2 phosphates become 2 ATP, and 2 NAD pluses become 2 NADHs. So the net yield, 2 ATP and 2 NADH. Here's where wording is important. If I ask you, what's the total amount of ATP produced in glycolysis? What's your answer? Yeah, 4. But if I ask you for the net production of ATP in glycolysis, your answer has to be 2. Okay, so when you're looking at a question, am I looking for total or am I looking for net? This is where the bits get confusing. So you have to understand what the question is looking for in terms of terminology. Read the questions carefully. This is what trips kids up. Okay? So the first half of glycolysis, when we're adding the endergonic phase, right, the energy investment phase, think of it as priming or pumping up the glucose. We're basically getting the glucose ready to split. We're going to phosphorylate the glucose. What kind of enzymes are involved in phosphorylation? How did we always recognize an enzyme that was not A's? That's just enzymes in general. Kinase. Yeah. Look here. Enzymes 1 and enzymes 2. Hexokinase and enzyme 3, phosphofructokinase. These are the enzymes, right? These two enzymes are the ones that are involved in the transfer of ATP. They're going to cause the glucose to be molecularly rearranged and therefore destabilize the glucose molecule. Okay, it can then be split here at step 4 and 5. Okay, the second half of glycolysis is the energy harvest phase. Okay, we're going to get NADHs produced. G3P donates hydrogens to NAD+, making NADH. That's going to oxidize the sugar and reduce the NAD+. Okay, we're also producing ATP. G3P is going to go to pyruvate. Okay, PEP here, phosphoenolpyruvate, is going to donate the phosphates to ATP. Okay, and this is what we call a substrate level phosphorylation. Okay, we're going to take, so we have DHAP here and G3P. Remember that the DHAP gets converted to G3P, and then we go through these phases. Okay, we're going to take a closer look at this PEP phase, this substrate level phosphorylation. Okay, so it's only here at the very end, these two steps, that we get some energy out of the system. Okay, let's look at substrate level phosphorylation. It happens twice. We're going to take a look at the second time it happens. Okay, in the last steps of glycolysis, where did the phosphate come from to make ATP? If you look at this diagram, to make ATP, where did the ADP get the phosphate from? Looking at this. Which thingy? Yeah, there's the phosphate. Okay, so this molecule is called phosphoenolpyruvate. Okay, so the phosphate came from the sugar, the PEP. Okay, this sugar is the substrate to this enzyme, pyruvate kinase. Okay, so here's what happens. Here's PEP with the phosphate, right? That's what this molecule represents. This purple blob is this enzyme, pyruvate kinase. And here's ADP sitting here. What's going to happen to that phosphate? What's this enzyme going to do to this phosphate? It's going to literally transfer it from here to here. Okay? So there's the PEP. There's the ADP. The PEP loses the phosphate, turns into pyruvate. The phosphate gets transferred onto the ATP, right? So it went from here to here. We now have ATP, okay? So the PEP became pyruvate, and the ADP became ATP, okay? So the PEP, the phosphate, transferred from PEP to ADP. We used a kinase enzyme, okay? And the ADP became ATP. Therefore, this, what do we call transferase phosphates? What kind of chemical reaction is a transfer of phosphate? Yeah, so there's the phosphorylation part of the name. What did we do? What did we use to transfer the phosphate? We used an enzyme and a substrate. So this is a substrate level phosphorylation. It's a transfer of phosphate that occurred using a substrate as a source of phosphate. That's what substrate level phosphorylation means. Production of ATP using a substrate as your source of phosphate. Okay, the PI comes directly from the substrate, Therefore, substrate level phosphorylation. This is important because there are, in fact, two kinds of phosphorylation in cellular respiration. This one, substrate level, and the fourth stage, which is called oxidative phosphorylation. Okay? Based on that name of oxidative phosphorylation, what do you think is involved in the transfer of phosphates in oxidative phosphorylation? It's not a substrate, obviously. There's no substrate in it. What's the word oxidative imply? Yeah, you're going to use redox reactions to phosphorylate ATP. Remember what I said before, earlier, that the entire point was to use an exergonic chemical reaction of redox to drive the synthesis of ATP, oxidative phosphorylation. That's stage 4. Okay, but before that, we make a little bit of ATP using substrate level phosphorylation. You should, by the end of the halfway point of this unit, when we finish cellular respiration, you should be able to describe to me the difference between substrate level phosphorylation and oxidative phosphorylation. Okay? So be sure that you understand these. If you don't, get in touch with me. Okay, energy accounting. Okay, we're going to use 2 ATP up to prep the glucose to break down. That's equivalent to the activation energy. Okay. When we do that, we're going to break the glucose down into 2 pyruvate molecules. That's going to produce a total of 4 ATPs and 2 NADHs. But because we use 2 at the beginning, our net gain is 2 ATP and 2 NADHs. Okay, so we had some energy investment here at the beginning. Okay, and we got a very small energy return at the end. Okay, and what we've done is we've taken 1 6-carbon sugar and we've converted it to 2 3-carbon sugars. Okay, so this is how we account for the energy in glycolysis. But as I said, this is inefficient. Okay, all of that, and that's all you get. Okay, about 2.2% of the energy. If that's the case, where's the rest of the energy that was originally in that glucose? Where's it sitting? Because obviously 98% of the energy didn't turn into heat. Where's the bulk of that energy? Some of it is, but we only got 2. Right? The amount of energy in NADH is like 131 kilojoules per mole. Yeah, the majority of the energy is actually still locked up in the pyruvate. Okay? So yes, glucose has so much more to give in the form of pyruvate. Because remember, we started with 2831 here. We've got about 260 kilojoules of energy here. And with the 2 ATPs, we got 62 kilojoules of energy here out of a total of 2,831. So most of the energy is still locked up as pyruvate. Okay, so it's not a lot of energy. But for well over 1 billion years, this is how life on Earth survived. Okay, there was no oxygen available in the atmosphere, so growth was slow and reproduction was slow, which is why prokaryotes evolved so slowly. Okay, total energy harvest, we're looking at about 3.5% of the energy in glucose. Okay, so if we could strip off more carbons, then we can get more energy out. Where's that energy sitting? Those carbons are sitting in the pyruvate molecule. Okay, so yes, for these organisms, it was a very hard way to make a living. Okay, now before we go on though, there's a bit of a problem that we have just looking at glycolysis, second half. Okay, we're going to go from raw materials, right, which is basically glucose and all of that, to final product, which is pyruvate. Okay, here's the net equation. Well, here's the total equation for glycolysis. Okay, glucose plus 2 ADP plus 2 inorganic phosphates plus 2 NAD pluses gives you 2 pyruvates, 2 ATP, and 2 NADHs. What's the rate limiting step in that equation? What will run out and cause the system to shut down in that yellow box? Not the phosphates. Because if you think about it, you get a net production of 4 ATP. As soon as those ATPs get used for something, there's more phosphates available. So the phosphates are not going to run out. Glucose is coming in from the environment. So all prokaryotes will have access to glucose. You're not going to run out of glucose. You make 4 ATP, though. You're always going to have ATP. What are you going to run out of? The NAD, right? Because eventually, the NAD plus goes to NADH. Once you run out of NAD plus, what happens to this step? Once all the NAD plus has been converted to NADH, what's going to happen to G3P? What's going to happen basically to step 7? It has to stop. There's no more NAD plus, right? This step, G3P going to BPG, can only occur if there's something to take electrons from G3P. So if there's no NAD plus, G3P cannot be oxidized, and the whole reaction stops. Okay, so eventually, if we don't do something, we're going to run out of NAD plus. Okay, so without regenerating NAD plus, energy production would stop. So question, how does an prokaryote that only has glycolysis make more NAD plus? Yeah, you have to transfer the hydrogen from NADH to something else. It's not going to be fat. Okay, so you have to remove the hydrogens from here so that you can regenerate NAD plus so that you can continue the second half of cellular respiration. Good guess, not fat, but good guess. So how is NADH recycled to NAD plus? It kind of depends on who you are. Okay, so you need another molecule to accept hydrogens from NADH. So you have pyruvate. If you are this, you're an aerobic organism. Therefore, what you do is this. You're going to shove it into the rest of cellular respiration, right? So you're going to go into the mitochondria, and we'll talk about all of this. But what this does is you're going to transfer, ultimately, the hydrogens in NADH to oxygen. Oxygen becomes water, right? And the NADH plus is regenerated for another round. That's if you have oxygen available, okay? But what do you do if you don't have enough oxygen when you're doing, like, say you're playing ice hockey? Okay, that's one method. If you don't have oxygen, then we do anaerobic respiration, also known as fermentation. But what kind of fermentation you do kind of depends. Okay, you can either do lactic acid fermentation or alcoholic fermentation. Those are the two options that we have. There are actually a couple others, but they're so specialized we don't talk about it in this course. Okay, so which pathway you use between these two depends on who you are. Yes, in part, Ibrahim, they are. So fermentation, which is anaerobic metabolism. So respiration without oxygen. If you are bacteria or yeast, you're going to take pyruvate and you're going to convert the pyruvate is going to take the hydrogens from the NADH, regenerating NAD+. The pyruvate is going to convert to ethanol plus a carbon dioxide, and the NAD+ will go back to glycolysis. Okay, so beer, wine, bread, things like that. Okay, but animals, all animals, and some fungi do something a little bit different. We do the same thing. Pyruvate's going to remove the hydrogen from NADH and convert it back to NAD, but we form lactic acid instead. Okay, that goes back to glycolysis. Okay, and we get cheese and anaerobic exercise out of it. Okay. Okay, alcoholic fermentation. Pyruvate, what literally happens, here's glycolysis, we get pyruvate. You lose a carbon dioxide and it becomes acetaldehyde. That acetaldehyde will pick up the hydrogen from NADH and become ethanol. Okay? So that's the process. The carbon dioxide, you're going to exhale out of your body. Exhale, you're going to get rid of its waste. Okay? This is a dead-end process. Why? So yeast at about 12% ethanol is going to kill the yeast. Okay. Why can't we reverse the reaction? This is why it's a dead end process. This chemical reaction is irreversible. Why? Not because the ethanol's stable. Okay. We built up ethanol. How do we get back to pyruvate? Because to be reversible, you have to get back to pyruvate. How do we get back to pyruvate? Count the carbons in ethanol, count the carbons in pyruvate. Yeah. Count the carbons. Ethanol has how many carbons? And pyruvate has how many carbons? 3. There is no way to get this 2-carbon molecule back to a 3-carbon molecule, because what's happened to the CO2? The CO2 didn't stick around inside the cell. It got removed as waste. Since it's been removed as waste, there's no way this ethanol can grab another carbon from anywhere to get back to pyruvate. Therefore, the ethanol sticks around, can't get back to pyruvate, and at 12%, it kills the yeast. Can't. Because there's no mechanism, but they don't have lungs. The concentration gradient is from the cytoplasm to the exterior. There's no active transport mechanism from the environment into the cell for CO2. It doesn't exist. Plants do something completely different. They're not, what they're doing is they use a different biochemical pathway called Calvin cycle. And they're using solar energy to form the formation of NADPH and ATP to drive the synthesis of sugar. Completely different process. Because there's no energy source because to add the CO2 to the ethanol, you need an energy source. Yeast don't have an energy source to do that. Okay. But if you look at lactic acid fermentation, okay, no oxygen present, this process is reversible. How many carbons in pyruvate and how many carbons in lactate? Yeah. Since they both have three, there's no carbon loss. So it is possible to convert lactate back to pyruvate. Okay. So once oxygen is available, the lactate goes back to your liver and is converted back to pyruvate. It's called the Cori process. Okay. And therefore you can get pyruvate back, and pyruvate then goes to the rest of cellular respiration. Right. And that's why, as Vedant said earlier, it burns when you first do this, right? And it's painful. And then that recovery period where you're breathing heavily, while what you're doing is your liver is now converting that lactate back to pyruvate, then all your mitochondria and your muscles are converting the pyruvate to carbon dioxide and regenerating ATP as fast as it possibly can. Okay? So pyruvate's a branch point. Okay? Without oxygen, you're going to do one of two kinds of fermentation, right? Either lactic acid or alcoholic, and that's anaerobic respiration. If you have oxygen available, then you shuttle everything down into the mitochondria, okay, and you do aerobic respiration. Bacteria, um, for the most part, are anaerobic. No, bacteria don't use alcohol fermentation. Well, some do. But for them, the remember the difference between yeast and bacteria. Bacteria are living in the environment, so the alcohol will just get washed away. Yeast, when we make alcoholic beverages, are trapped in a bottle. That alcohol doesn't get washed away, so the concentration increases till about 12%, which then kills the yeast. If the yeast lived in the wild, right, fermenting an apple in an orchard, yeah, the environment would wash away that excess alcohol and they could continue to survive. But because we bottle them up, that alcohol can't escape. So here's a question for you. How do we get grain alcohols? How do we get rum, rye, whiskey, 30, 40, 50 overproof alcohols? How do we get those if yeast dies at 20%? Yeah, how do we concentrate it? What's the process called? Yeah, distillation, right? That's what an alcohol still is. That's how we get our grain alcohols. Okay, no, not beers. Beers are highest percentage in beer I know is probably about 8%, which is enough for the yeast to survive. But grain alcohols, yeah, rum, rye, whiskey, vodka, moonshine, all of those are going to use distillation. Okay, 10-minute break. It's 10:10 basically. So we'll take a break for now, and we'll start stages 2 and 3. All right, so we'll see you in a bit. I'll be ready in a couple of minutes. I'm just checking out an ETC video that I may use with you guys. No, I won't. It's 16 minutes. It's way too long. I've just started marking the other class, and I've gotten through 5 of the other classes so far. So hopefully I get through them. Hopefully by the end of the week, but we'll have to see. I mean, you're the bigger class. I have 32 of you guys and 27 of the other. So it's going to take a while for me to mark these, which is why I don't mind giving the osmosis paper extensions. Okay, before going on, I'm going to give you an overview on glycolysis. There's a 3-minute YouTube video on it, which is pretty good. So I'm going to let you guys watch that, and then we'll jump into stage 2, 3. Let me take a look at it later.
ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3-carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP, and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, Glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
Okay, let's take a look at this. Copy link address.
Hey, sugar. Come on over here, sweetie. Let's break it down. Glycolysis is a series of reactions, enzymatic actions, energy transactions. Takes glucose, a molecules of sugary, breaks it down to NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes. Organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis, come on sugar for the breakdown. For the breakdown. Investments, like striking a match. That energy you put in makes the fire catch. For glycolysis, investments, 2 ATPs, which act as activation energy. Enzymes take phosphates from ATPs, jam them on a glucose, rearranging it to fructose, leaving fructose 1,6-bisphosphate on the table with 2 phosphates. It's highly unstable, moving us to the 2nd phase, the cleaving of fructose bisphosphate, because glycolysis means splitting sugar, you can see. Cleavage yields 2 molecules with carbons 3. One is glyceraldehyde 3-phosphate G3P. It continues on our pathway broken down for energy, but the second one, an enzyme, will immediately convert into a second G3P. Glycolysis, come on sugar for the breakdown. For the breakdown. Phase 3, G3P gets rearranged and oxidized by an enzymatic assembly line that harvests energy from each G3P, 1 NADH and 2 ATPs. Double this yield per G3P to 2 NADH and 4 ATPs. That's the gross yield for every glucose in. A generous accounting of glycolysis' win, but 2 ATPs were invested in phase 1, so your net just 2 you can use to jump or run. Put 2 in, get 4 out, your net gain is 2 ATPs that you can use. If that doesn't seem like very much, it's 'cause it ain't. There's tons of energy left in pyruvate. The 2,3 carbon molecules we're left with at the end, and what happens to pyruvate's going to depend on the metabolic pathway where pyruvate gets sent. If it's anaerobic, it'll be fermented, but in aerobic cells, pyruvate's termination will be the Krebs cycle and total oxidation. Glycolysis, come on sugar for the breakdown. For the breakdown. Glycolysis starts with investment of 2 ATPs to the glucose that we started with. The product is cleaved into 2 G3Ps from which the cell harvests NADH and ATPs. This anaerobic pathway is respiration's first phase, billions of years old, evolved in ancient days before O2 accumulated in the seas, before eukaryotic cells arrived on the scene. It's everywhere, ubiquitous in every organism. Bacteria, sequoia tree, no matter your metabolism. Happens in the cytoplasm, doesn't need no organelles. You want to find glycolysis? Look in any cell. Glycolysis, come on sugar for the breakdown. For the breakdown. Glycolysis, come on sugar for the breakdown. For the breakdown.
built custom industrial machines in days.
Oh no, there's a funny comment on here. I failed my exam because all I remembered was the glycolysis, come on sugar for the breakdown part. Oh, that's amusing. Okay, I did put that up there if you want it. I must admit I like his ETC stuff better, which we'll be looking at later. And I'm also going to put on a bunch of, I'll put up a section of just music videos that are related to this stuff later in this unit, like in this section. Okay. And a few other things. Okay, pyruvate oxidation. So this is stages 2 and 3. So this, just share it. It's not sharing. So this is Krebs cycle. And you can grab that handout if you like, or you can take a look at it in your textbooks. Your textbook version is way more complicated than this. So this is a simplified version. I'll leave it up to you. Okay, so we're starting 05, pyruvate oxidation in Krebs. Load up. Okay. No worries, Teja. We've been just watching some singing videos, so you didn't really miss on anything yet. I haven't started the next section. Waiting on Doc School Kong. Okay, so this is what we've been looking at up to this point. Okay, we finished glycolysis. We're at pyruvate. Okay, so what we're going to look at are stages 2 and stages 3. Stage 2 is called pyruvate oxidation, and stage 3 is Krebs cycle. A couple of points I'm going to put in the chat. So pyruvate oxidation is also known as transition step. Okay, so there are 2 terms for this stage: pyruvate oxidation or the transition step. They both mean the same thing, stage 2 of cellular respiration. The other thing is, depending on who you look at, some sources combine steps 2 and 3 and call it all recycle. Others separate as 2 stages. Okay, so keep that in mind when you're reading something outside of class. In class, I will always refer to it as 4 stages: glycolysis, pyruvate oxidation, Krebs cycle, oxidative phosphorylation. But there will be some references in stuff that you look up later that will basically say glycolysis, Krebs cycle, oxidative phosphorylation, and only this 3 stages. Okay? There are, in my mind, 4 stages, but it depends on who you use as a source. Please be aware of that. Okay? All right. So, as we've already stated, glycolysis is only the beginning of the entire process. Okay? Pyruvate itself has more energy it can yield. You've got 3 more carbons you can strip off or oxidize. If oxygen is available, it's going to enter the mitochondria, and then the enzymes of Krebs cycle are going to complete the full oxidation of sugar to CO2. Okay, so we're going to go from a 3-carbon pyruvate to 1-carbon, 3 1-carbon CO2s. So this is what cellular respiration is. Okay, so step 1 is here, glycolysis. Step 2 is this arrow. They don't list it in this diagram, but that's transition step or pyruvate oxidation. Then Krebs cycle, stage 3, and electron transport chain, stage 4. Okay, you notice that stages 1, 3, and 4 produce ATP. Stage 2 does not. That's the big takeaway from here. Okay, all right. First thing we have to look at when we look at this, we have to look at the structure of the mitochondria. Okay, so the mitochondria itself is a double membrane energy harvesting organelle. It's got a smooth outer membrane, okay, and a highly folded inner membrane. That inner membrane is called the cristae. Okay, in between the inner membrane and the outer membrane, or the outer membrane and the cristae, is a fluid-filled space, which is called the intermembrane space. Okay, the fluid area inside the cristae, this area here with the dots in it, is called the matrix. It's an inner fluid-filled space. Think of the movie The Matrix, where power generation occurs. The matrix contains mitochondrial DNA, which is a black loop. Okay. It also contains ribosomes, so it can make some proteins, so mitochondrial proteins, which are the black dots. And then there are enzymes, both in the membrane and free, floating around free in the matrix. Okay. So that's the general structure of the mitochondria. Okay. And as of electron photon micrograph, this is what the mitochondria would look like. Okay. It kind of makes sense in terms of endosymbiosis. I mean, if you look at the mitochondrial DNA, it's a loop, right? Just like bacterial DNA. And it contains the components similar to a bacterium. So it makes some sense that this at one time may have been a free living bacterial organism that was co-opted by endosymbiosis into an organelle. Okay, more diagrams, more photomicrographs of mitochondria. So what cells in your body do you think would contain a lot of mitochondria? Which ones specifically? No, skeletal have a few, but there's a type of muscle that has a lot more. What muscle moves continuously? Yeah, cardiac. Okay. What other organ would be high oxygen demanding and therefore a lot of mitochondria? Brain. Yeah, heart and brain are the two that have the maximum amount of mitochondria. Third one might be brown fat, which we'll talk about later. Okay. Mitochondria function. Okay. So they have these membrane-bound proteins, which are enzymes and permeases. Roughly about that, yeah. Okay. So a permease is basically a molecule that allows certain polar molecules through the membrane. Okay. And so you need them in order to set up an electrochemical gradient that we'll talk about later. Okay. The reason that the cristae itself is highly folded, more surface area for the enzymes and permeases to exist in, so that you can pack more of these things into this area. Okay, so form fits function. Okay, so step 1 or stage 2 is the oxidation of pyruvate. Okay, the reason that we need to modify pyruvate in order to complete the rest of cellular respiration, pyruvate itself cannot cross the mitochondrial membranes. Okay, pyruvate is impermeable to the mitochondrial membranes unless we modify it, which is why stage 2 must occur. So stage 2 is a 3-step oxidation process. Okay. It's going to release 2 carbon dioxides because there are 2 pyruvates, right? So there's going to be 2 of these. It's going to reduce NAD plus to NADHs. So you're going to start with a 3-carbon molecule. It's going to become a 2-carbon acetyl coenzyme A and 1 carbon here. The NAD becomes NADH. Okay, so if NAD becomes NADH, that means there had to be a redox reaction step that occurs here. And we also produce acetyl coenzyme A. What happens to the carbon dioxide? We're going to exhale it and it's going to leave your body. That acetyl-CoA is what enters Krebs cycle, stage 3. Okay, so let's look at this step. We start with the pyruvate molecule. In order to get the pyruvate into the mitochondrion, there is a transport protein involved. This transport protein is going to strip this this part of the molecule, CO2 here, out. And that becomes the carbon dioxide that's going to leave the cell. Coenzyme A, which is basically a cofactor, or a coenzyme rather, is going to come in and it's going to pick up this remaining acetyl group and become acetyl coenzyme A. That's where this molecule comes from. Okay, the coenzyme A acts as a shuttle between stage 2 and stage 3. Okay, the CO2, as it goes through, as pyruvate goes to acetyl CoA, right? 3 carbons becomes 2 carbons. 1 carbon has to be released to CO2. So this is an oxidation step. Whenever CO2 is coming off of a carbohydrate, we have an oxidation, which means we got to have a reduction, something to accept the hydrogens, which is NAD plus going to NADH. Okay. This happens once for 1 pyruvate. So 1 pyruvate produces 1 CO2, 1 acetyl-CoA, and 1 NADH. Since there are 2 pyruvates, because glucose splits into 2 pyruvates, there will be 2 acetyl-CoAs, 2 NADHs, and 2 CO2s. Okay? Be aware of your numbers. Am I asking what happens to a single pyruvate? Or am I asking what happens to the glucose equivalent? Okay? Because if you give me pyruvate numbers for the glucose equivalent, you're only going to have half the numbers and you'll get it wrong. Okay? Be aware of what numbers I am looking for. Okay, stage 3, Krebs cycle. Discovered in 1937, he worked out the entire Krebs cycle. 1953, he won the Nobel Prize in medicine for that discovery. Okay, Krebs cycle is also known as citric acid cycle. It's also known as the TCA cycle. TCA does not mean the citric acid cycle. TCA cycle equals the tricarboxylic acid cycle. The reason it's called the tricarboxylic acid cycle is because it releases 3 carbon dioxides in the process, if you include transition step. Okay, it's an 8-step pathway that catabolizes a 6-carbon citrate molecule. Okay, we'll talk about where the citrate molecule comes from in a second. It evolved later than glycolysis. Does that make evolutionary sense? Yes. Bacteria evolved about 3.5 billion years ago, which is when we have glycolysis. Free oxygen started increasing in our atmosphere with the evolution of photosynthesis, which is about 2.7 billion years ago. The origin of eukaryotes started about 1.5 billion years ago, which is when aerobic respiration came into being, production of organelles, and mitochondria. So without mitochondria, you can't have Krebs. Therefore, it makes sense that this would have happened after glycolysis. Okay? Now, so early bacteria would have used glucose in glycolysis. Well, there were also capable of using inorganic chemicals like hydrogen sulfide, so sulfur-using bacteria, which still exist today. There are those that would use methane gas. So think those organisms, the methanogens. Yes, exactly. Those would have existed. And remember, photosynthesis, as we're describing it, we're talking about the green cyanobacteria. But there were other forms of photosynthesis. Our oceans initially were purple in color because there were, they didn't produce O2, but they used a pigment that could capture purple light, yeah, of a form. And so could use purple light as a source of energy for a form of photosynthesis, which was not chlorophyll based. And they would then make complex organic molecules as well. So for a while, our oceans were purple in color. The reason that we have green bacteria and photosynthesis is green is because when photosynthesis initially developed, all the purple and blue colors were being absorbed by the purple bacterium. Only the deep greens or the reds made it into the oceans, so deeper ocean colors. Therefore, yeah, the cyanobacteria developed green um, pigments in order to absorb the remaining reds and oranges left in the water that they could photosynthesize. Because the blues and purples were no longer available, the purple bacteria already had it. It removed it from the water column. So NASA exobiology researchers confirmed their oceans were once rich in sulfides that would prevent advanced life forms, such as fish and mammals, from surviving. Evidence of photosynthetic bacteria that require sulfide and sunlight to live, known as purple and green sulfur bacteria, because of respective pigment colorations. These single cell microbes can live in environments where they simultaneously have access to sulfide and sunlight. They also found low amounts of fossilized remains of algae and oxygen-producing cyanobacteria. The relative scarcity of these organisms is due to poisoning by large amounts of sulfide. Right? I mean, remember, Emma, we still have purple, or not purple, well, some purple, but we also have like pink and other bacteria today, right? Right? These are the pools of the halophile bacteria. So things like this. All kinds of bizarre colors. And they're these colors to protect themselves from the intense sunlight, right? The UV radiation. Okay. Actually, let me see if I can find it because it's kind of neat. I haven't seen this video, so we'll see.
It's a digital writing assistant that you can download as a browser extension. It is extremely helpful.
Picture the view of Earth from space. Beautiful, right? What colors do you see? Besides the blue of the oceans, the dominant color of our planet as we know it is green. But imagine a time when the Earth looked a little purple. All that green we see today is because of chlorophyll, the pigment that plants use to harness the sun's energy through photosynthesis. Chlorophyll looks green because it absorbs the blue and red parts of the visible spectrum to use in photosynthesis, while reflecting green light. But the fact is, the sun actually emits more photons in the green wavelengths than any other part of the visible spectrum. And this has biologists puzzled. Why didn't living things evolve to take advantage of all that literal green energy by absorbing green light instead of reflecting it? Well, scientists think that green light is so plentiful that absorbing all of its energy would actually be harmful, causing damage to the pigment, similar to a sunburn. But another more interesting explanation has been proposed too. It's called the purple Earth hypothesis. So the idea goes, maybe the very first photosynthesizers on Earth didn't use chlorophyll at all. Instead, maybe they used another simpler light-sensitive molecule, one that did absorb all that abundant green energy and reflected purple light. This idea is at least plausible because such tiny lavender forms of life exist on Earth today. They're called halobacteria. Halobacteria aren't actually bacteria. They're archaea, single-celled organisms that mostly thrive in extreme environments where almost nothing else can live. For example, they're known for living happily in concentrated salt solutions. Their membranes contain a light-sensitive pigment called retinol, which absorbs green light, making it appear purple as a result. Retinol is a simpler molecule than chlorophyll, and it's easier to produce while making the most of that abundant green light. So the purple Earth hypothesis suggests that back in the Archean eon, before chlorophyll was even a thing, Earth's oceans may have been dominated by microbes that were a lot like halobacteria, ones that used retinol or some other purple pigment to harness the sun's energy. This idea was first proposed in the mid-2000s by microbial geneticist Shil Desarma at the University of Maryland. He says his hypothesis might help explain why today's photosynthesizers don't absorb the green light from the sun, because they adapted to a world where an abundance of other organisms were already monopolizing it. According to the purple Earth hypothesis, chlorophyll eventually evolved in a different competing lineage of microbes to take advantage of the wavelengths of light that purple archaea weren't using. Picture a mat of green chlorophyll-based microbes underneath a raft of purple retinol-based microbes, soaking up the leftover dregs of light. In this scenario, halobacteria, or something like them, could have been among the earliest forms of life on our planet, and their world would have been very different from the one we know today, hot, bombarded by UV rays, and rich in sulfur and methane. Unlike today's chlorophyll-using photosynthesizers, these organisms wouldn't have produced oxygen. In fact, they would have thrived in the oxygen-deprived environment of early Earth. That is, of course, until those lowly chlorophyll producers started taking over. Chlorophyll is a more complex molecule than retinol, and it doesn't soak up those abundant green wavelengths of light, but it is more efficient, making better use of the light that it absorbs and converting more of it into usable energy. And in the long run, that's what may have mattered most. Of course, this is all speculation. The fossil record is pretty limited when it comes to microbes from billions of years ago, so the purple Earth hypothesis can't yet be proven either way. But it does fit with what we know about the atmosphere of the early Earth. For instance, we know that around 3 billion years ago, there was barely a trace of oxygen in our planet's atmosphere, so there couldn't have been a lot of chlorophyll-based photosynthesis going on. But we also know that about 2 billion years ago, some microbes that did use chlorophyll, like cyanobacteria, came on the scene and began to flourish, releasing tons of oxygen. That flood of oxygen would have killed off many of the simpler, possibly purple microbes that came before them. This microbial carnage came to be known as the Great Oxygenation Event, which we've talked about before. But if Desarmes' hypothesis is right, then why are there still halobacteria and other archaea around today? Wouldn't they have been wiped out? Well, his work suggests that some crafty halobacteria may have managed to snatch a few genes from the DNA of other microbes that allowed them to survive in the presence of oxygen, which has helped them persist into the present. Again, it's all speculation, but a purple Earth is fun to think about. And if this idea is true, it could also have big implications for the search for life on other planets. One way we seek out other living worlds is by looking for planets that reflect less red light than we'd expect. This could mean they have organisms with chlorophyll that are absorbing those red wavelengths. But if life can evolve with more than one way of making energy from light, maybe we shouldn't just be searching for planets that are green like ours. Maybe we need to be looking for purple worlds as well. What do you want to know about the story of life on Earth? Let us know in the comments.
It's now 10:55, so we'll stop there. We'll continue this tomorrow. All right, and you are dismissed. Have a good one.
Thank you. Have a good day.
You too.
Bye, thank you.
I'm still online if you need any assistance. I'll be marking tests.